Thank you, everyone. I'm very happy to be here today. And thank you to the organizing committee, to Yelvi and to Jessit for putting such an amazing program together, and to Diana and Say that are always on site helping everyone, but yeah, like all the steering committee. And thank you, everyone, for being here today. I think this is one of the biggest IACB Latin America conference we have ever had. So today I will share some of the work we have been doing in Mexico and in Latin America towards setting up the infrastructure to really build frontier research in our region. So I just want to talk a bit about patient registries. So as you probably know, patient registries are organized systems that collect, store, and analyze information about individuals living with a particular disease or condition. I think some of the main resources that are well known is the All of Us UK Biobank and, for example, the Twin Research Australia. They are very useful for clinical research, but also they help empower genomic and genetic data research because, well, you have a lot of metadata on these people because they were registered for clinical research as well. So this is a plot that probably you already saw several times about the diversity in genetic research and genetic and genomic studies. And you can see that Latin America is really poorly represented there. So we are usually taken into account as Hispanic. So all the region is put together in one bag, and then is analyzed all together. But this makes up, even putting all the people together, it's less than 2% of the genetic data that is out there. But we know that our region has a lot of great diversity due to constant migration, difference in the ecosystems, but also because we have different marks in the Native American genomes that inhabit our countries. So these factors have shaped population with diverse genetic backgrounds in which genetic variation manifests in unique effects on complex strains, including the immune system. So that's also very important. The immune system is the thing that is reacting to our environment. And we have diversity in genes, but we also have this diversity in the environment. So this is very interesting. And this means we also need functional genomics. When we actually see into the functional genomic representation of different ancestries, we see that most of the time Latin America is not even existent. So we are not part of the cancer cell lines, we are not part of the EQTL catalog, and we are not represented in the TCGA project. We are barely represented in the EWAS project, and we make up less than 2% of the data sets. So genetic data is bad, functional genomic data is worse. So what do we have in Mexico? So we'll focus on Mexico because this effort started there. So these are the two major genomic resources in the region. We have the Mexico City Prospective Study, which is a survey based in two regions of Mexico City, Coyoacán and Iztapalapa. And Coyoacán is middle upper level class and Iztapalapa is low middle class. They did some medical survey with anthropomorphic measurements. They exome sequenced 150,000 individuals. That's a lot of people for Latin American studies. The recontact is based on the address, which is a challenge because Mexico City is heavily gentrified at the time. So a lot of people no longer live in these places, which makes recontact very hard. 10,000 individuals were recontacted for genetic studies in 2015 and 2019. There is a heavy focus on diabetes, cardiovascular diseases, and smoking outcomes. Again, these projects started in the 90s. We had different questions at the time. So we are just sequencing what we had, what we collected with the data we had at the time that was interested at the time, which, like, smoking outcomes are important, but we can already learn we shouldn't smoke. That's something that is settled. Probably analysis is going to be helpful, but the issue is that we cannot go back and ask them, what happened in your life? How did you get sick? Which is a challenge. The major publication came in 2022. And one of the things that is to be commented and noted on this project is that while it is led between Oxford University and the National Autonomous University in Mexico, UNAM, the data lives in UK. There's not even a copy in Mexico. So it is totally controlled by Oxford University. They are open for Mexican scientists to analyze the data using their infrastructure, which is always a challenge because the connection of internet, because you have to go through a lot of paperwork to actually get it. And this is Mexican data. So you can already see this is something that needs to be noted and needs to be improved for future projects. The second major resource is the MEX Biobank, which is using samples from the National Health Survey in the 2000s. They collected a big survey on health status, but they also did blood drawing. They already generated genotypes data for 6,000 individuals across the country. They have 4,000 samples in the fridge. And they have these general health surveys. There is no recontact. There is no way to identify these people. There is no way to recontact them. We cannot ask them any further questions. So one of the things that is not non-existent in both resources is any questions about immune diseases. There's nothing about autoimmune whatever, and there's definitely nothing about mental health issues, because that's something that was not important in the 90s. We were really a bit more worried about diabetes and smoking. Depression, that was not a thing. Anxiety, no one cared. The preprint sorry, I actually had to update this. They had their major publication earlier this year, and this is work led by Mexican institutions, mainly by Langevio, and the data lives in Mexico. So we have the capacity in our countries. I know there are researchers in Latin America that are able to handle this level of data. So we can collaborate with other institutions abroad and get their help to improve our science and get it further, but we can also work on it. We can be part of the leading team. So some challenges of population genomic studies in Mexico. There's no way we can use electronic clinical data because there is a big diversity of electronic medical records in Mexico. We have 4 independent health systems coexisting in the country. So, and actually, if you change out, you could change system, and your data gets erased in one, and you have to start all over again in the other one. A complete mess. And actually, the databases storage systems greatly hinder data sharing and interaction among health institutions. There's no way they can communicate. And actually, a lot of the databases get erased when the governments change. So that's impossible to work with. And effectively, we have 33 million people with no access to medical services, but actually half of these people is not that they don't have the insurance to access the hospital, it's that there is no hospital to be accessed. So that's also a challenge. The recontact, you could see that was not part of the design of our major resources. And there is a heavy issue of decentralization. So most of the projects are heavily focused in Mexico City. And actually, when you are working with any disease, you are usually working with one of the national health institutes, which are all based in Mexico City. While they provide service to all the country, the people need to be in Mexico City to receive the service. So that, at the end, really narrows it down to people that live in Mexico City. There's a challenge in collaborating and engaging with the medical community. And this comes because, well, we don't have a lot of resources for health. So medical services are usually focused on helping people live rather than doing research. So that's always a challenge, engaging them, making them being part of the research, because they don't really have the time. It's not that they don't want to, they are interested. But when you have 15 minutes appointments for your Parkinson project, for your Parkinson patients, that there's no way you can plug in a 30-minute survey for a Parkinson research project. So it's a big challenge. There's also a lot of lack of trust towards the scientific community in the country. This is well documented by INEGI. So most of the people actually distrust the scientific community. They feel that if they give us their data, we won't use it correctly. So convincing people to participate in science is a challenge with this environment. So how do we deal with this? So we created a Mexomic consortium, and this consortium is developing a comprehensive medical history surveys. We have ethics and consent that contemplate the option to be recontacted for follow-up surveys and analysis. The ethics are approved for biobankings because sometimes you have the money to start the surveys and the database, but you don't have the money to sequence. And there's really little chance that you will find these people again in the future. So you rather just have the sample at the time. We include mental health cognitive assessments and lifestyle surveys. And we really have a focus on nationwide recruitment perspective focus. And we work a lot in public engagement through continuous communication with the participants. And this is very important because if we want to start building this trust that people want to be part of research projects and participate in them, we have to improve our communication with the people. So we started in 2018 the Mexican Twin Registry that now has over 5,000 people that were born in multiple births. Now, then in 2021, we launched the Mexican Network for Parkinson Research that now has over 1,000 individuals registered with DNA samples. And we also started the Lupus Mexican Registry, which I will talk a bit more about later today. And all our methodology was recently published. And one of the things I want to highlight is that this is very low cost. So this is designed for our countries where you really literally have only like 3 pesos and you have to be able to work and start building the infrastructure. So if you are interested in building this type of resources, I think this is a very good way to start, and we are open to discuss and share everything we have built up to now. So going into lupus, lupus is a chronic autoimmune disease. It's multifactorial, multisystemic, and heterogeneous, which makes diagnosis very hard because the same patient can have really different phenotypes across their lives. So it's very hard to diagnose it. Some people can have skin issues, some people can have joint issues, and the same person can have both. So it's very hard. It mostly affects women, 9 in 1, and most of the people that get diagnosed are between 20 to 40 years old. So it's a very challenging time in life where you are becoming productive, when you're finally finishing school, when you want to start a family. And this is the moment when you get this diagnosis. So it heavily affects people. There is a calculation of prevalence in Mexico that goes between 20 to 80 cases in 1,000 individuals. I will talk a bit more about this. And we know that it's a disease that is affected by the environment, the socioeconomic status, and the genetics. And we know that ancestry influences the manifestation and severity. Where African, Asian, and Hispanic have higher prevalence, mortality, and severity of the disease. And when we see the prevalence map in the world, you can actually notice that Mexico doesn't even have data, regardless of the fact that we know that Latin American people and actually Mexican individuals have a higher prevalence and also develop a much more aggressive form of the disease. So this is what we know in Mexico. We don't have epidemiological surveillance in place. We know that it affects mostly women as well in our population, and this prevalence, the 21,000, is something that is documented in the health institutes, but if you look for the reference on how it was calculated, you cannot find it. It looks really like someone imported the data from the United States. But there are some studies across the country, and you can see that the least prevalence is 41,000, depending on the region. And that's already something that is important. It's not the same being from Oaxaca that is in the south, that being from Nuevo León that is in the north. So we can tell already that maybe the Native American component is something important in this disease. So why do we need a lupus registry in our country? The Mexican Lupus Registry arises from the need to have a source of information on the characteristics of Mexican people with lupus for what we lack epidemiological surveillance. Knowing these characteristics and understanding how lupus affects our population is going to help us improve monitoring, improve diagnosis and treatment, and will also open avenues for research. So we launched this resource in 2021 in May, and we have a very big component of public engagement. We work with the patients since the beginning. They have been key part of the design of the surveys, of how we communicate the research. So this is very important, and we think this is different from what we usually do. It takes time, it takes a lot of time and resources, but it's worth it because the patients are not just the focus of research, they are part of the research team, which is different. So we now have 20 rheumatologists that also joined the recruitment of patients, and we started recruitment, it was 2021, so it was still COVID. There was no way anyone was going to the hospital or to the MD ever. So we really started through social media with self-assessment reports. But now we work as well with rheumatologists that directly input the data on the clinical manifestations. This is a team. You cannot do this yourself. This is all the institutions that are part of this research. So it's mind, it's leak, but it's also Sanger. So again, it's not that we don't want to collaborate with institutions abroad, but we want fair collaborations. And this is something that I have found with Sanger. We also work with Boston Children's and with QIMR in Australia. But you can see most of the institutions and within Mexico, like the Health Institute for Social Security, the Nutrition Institute, and the Psychology School as well, and the Rheumatology College. And those four purple logos are the community of patients. So those are associations built by the patient that are sitting in the board of the registry, that are being part of the research, and that also have their own questions, and they also have their own requirements, and they want to be part of that. So this is a summary of our progress. Through community engagement, we have reached over 7,000 people. We have successfully been able to register 2,873 people. We have social demographic data for 1,700. And you can see the coverage across the country. There are really few regions where we're lacking people, but you can also overlay the presence of clinicians, of rheumatology clinicians with the map, and it's perfectly like a copy. So you can see that you have the least individuals with the disease in the places where you don't really have someone to diagnose the patients. So that's a big challenge as well. And we have mostly women, and most of the people are between 20 to 40 years old. I apologize that ended up being too small to be seen. Most of the people have university studies, and a bit of half of them are currently employed. And regarding clinical characteristics, most of the patients have alopecia or damage in the skin as well. And again, I apologize because it ended up being smaller than I expected. And we also registered their treatment. So most of them take glucocorticoids and antimalarics, but there are other people with other type of treatments. And currently for biobanking, we have biobank 164 samples for DNA, and for which we have the whole genome sequence for 87 SLE patients and 10 controls, and we have biobank PBMCs of 97 patients and 5 healthy controls. These healthy controls are a bit different from what I will talk later, because this only means they don't have lupus, not that they don't have another disease. But later I'll talk a bit more about that level of healthy. So one of the things that was very important for the patients was to have documentation about their quality of life effects. So how the disease affects their quality of life. So this was assessed through the OMS, through the WHO quality of life assessment. And one of the things I want to highlight is that by building this consortium, we actually were very careful to share surveys across projects, which empowers everyone's research, because the control, like one population can be used as a control for another study. So that really brings and helps taking into like the most advantage of the research we are building. So here we took as control group female twins, obviously one twin per set, because otherwise you will have mostly the same data. And we compare with the lupus patient. We also had a second lupus control that were friends from the lupus patients. And we compared the quality of life perception between patients and controls, and you could see that is lower. So this disease heavily affects the quality of life in different spectrums. This is a way bigger study. And this was something that was requested by the patients, because they really needed to present this to politicians to change some things in the health law, because they want to introduce lupus as one of the inhabilitating diseases, so they will have more time to go to the doctor and have more resources to get their medication. So this was an analysis that was built, was driven by patients' interest. So having this research really opens avenue and potentializes research, because we have data and we have samples. So one of the things is this demographic characterization, sorry about the typo. We can also help us analyze mental health studies. So now we're currently analyzing the effect on the disease on depression and anxiety. We are also doing now brain imaging. So we currently have 30 MRIs in some patients. We also are assessing treatment effects in the health perception. And we will as well assess disease progression and damage based in different surveys, but we also empower genomic and genetic studies with this resource. So what are we doing in this regard? So there are two main avenues we are taking here. So one is the gene expression profiling of tolerogenic dendritic cell differentiation, and the other one is single cell mapping in lupus patients. So for the characterization of dendritic cell tolerogenic induction in SLE patients, so why do we want to do this? So dendritic cells are antigen-presenting cells. So basically, these are the cells that trigger the immune response. But there is a type of dendritic cells that is called a tolerogenic dendritic cell that actually is able to stop the immune response. So in general, people, when they start having self-antigen recognition, at some point they will build self-tolerance. So even if you have cells that are dying, that are your cells, your own immune system will say like, oh, okay, I shouldn't attack that because it's myself. So that builds self-tolerance. The issue is when you lose this tolerance, then you have lupus and your body starts attacking itself. So what we are proposing, and that's actually one of the potential immunotherapies in lupus and in other autoimmune diseases, this has been tested in diabetes and in rheumatoid arthritis, is that tolerogenic dendritic cells can stop the immune response through toning down the response from Tregs, and then, well, you won't have the inflammation. So what are we doing here? So we sampled 23 patients and 10 controls. We purified PBMCs, and we took only monocytes, which we differentiated into monocytes, into dendritic cells that come from monocyte. That's why they are called MO disease. And we have the tolerogenic dendritic cells as well. So we want to compare them and see whether they are different between patients and controls. Spoiler alert, there's no difference. So at the beginning, this was like super sad, no? There's no difference between patients and controls in their gene expression between dendritic cells, tolerogenic dendritic cells, and non-tolerogenic dendritic cells. So we thought that, well, we can keep analyzing the data and see what we get. And one of the things is that what we were analyzing was the change pattern between what changes between being a monocyte and a monocyte dendritic. Sorry, it should say monocytes here. So what changes to becoming a monocyte to becoming a dendritic cell, and what changes from becoming a monocyte or becoming a tolerogenic dendritic cell? So in this case, we see different numbers of genes, and there is a difference in the genes that change between this differentiation to this differentiation. And we find 944 genes that are different between this path. Oh, sorry, I keep going on. Between this path and this path. But I won't keep spoiling this story because my PhD student, Ana Hernandez, is going to present it in a talk this Thursday at 2 p.m. So please be tuned and come to that talk if you want to know what happened here and which were the pathways that we were able to identify. So in the other story, what's going on with the lupus single cell characterization? So at the time, we're still in the sampling phase. As I told you, we have 97 PBMC samples collected. You can see that the coverage is a bit lower. And this is because we need to draw blood from people, which is in Querétaro, which is the pink part where we have most of the patients. But there is a lot of migration towards Querétaro, so that's why we have representation in other regions. We do want to increase this, but at some point, getting patients is hard, so we don't turn anyone down. The age distribution, again, is really between 20 to 40 years old. And we mostly get women, but we do have some men that have been collected and are part of the study. And we will be opening new sampling sites in Monterrey, that is in the north, and in Tapachula. And as I was telling you, for example, Monterrey was the place with the lowest prevalence calculated of lupus. And Tapachula, well, this is Oaxaca, but Tapachula is Chiapas, which is near. Someone will kill me because it's not actually the same indigenous population, but at least it's closer. So we foresee that this is going to be a more interesting population for lupus. So this is work that I actually started with Gosha many years ago, and it's slowly coming into shape. And if you are interested in her lab's work, she has a keynote on Thursday at 4:30 p.m. So But for analyzing patient data, you actually need control data. This is very important. And for this, we started Project Jaguar, where we are aiming to map immune cell diversity across Latin America. And our aim is to address how genetic diversity shapes immunity. Again, immunity is really the thing that is responding to your environment. Our environments are different, and we have to acknowledge this. And our diversity is very rich in the genetic factor as well. So this is a strength. We have environmental diversity and genetic richness in our region. How are we going to do this? Using single cell. So we are going to produce single cell RNA-seq data, single cell attack-seq, and single cell site-seq data. If you don't know, site-seq is proteomic data, so that's the membrane proteins. And we want to see how diverse ancestry impact gene expression and the composition of the immune system. This requires a super big team, and really these are stars. So from Brazil, we have Benilton and Heitor. In Colombia, we have Jessit, that is also here. So if you're in Colombia, you want to work in this project, go reach for Jessit. In Peru, we have Luis, and in Chile, Marce. In Argentina, Danilo and Pablo. In Uruguay, Maximiliano. And in UK, because we needed the data generation expertise and also the infrastructure, we're collaborating with Sanger through Gosia Trinca and her team. So everyone that is in pink is around. Benito actually just left, so sorry that I told he was here just now. But Jessit is here. You can see poster from Luis's lab, a poster from Marce's lab as well. And you have Danilo as well around here if you're interested. So our methodological approach, so we are collecting blood PBMCs from 1,080 volunteers. So we have a system based on the Mexomics data management and on the surveys that we developed through that consortium, mounted on RedCap. We have the conserved forms, the complete survey with metadata on lifestyle, socioeconomic status, and environmental factors, and we do the PBMC collection only on healthy individuals. And this is very healthy people. So, well, we call it disease-free because we really have a systematic survey on a lot of diseases, and they have to say no to everything. So this is a very difficult population to get. We collect the PBM samples, we process and storage them in Latin America, then we ship them to Sanger, and at Sanger, they process them, and they try to enrich cell types, because this is very important, otherwise you end up with only T cells. And then we will produce RNA-seq, site-seq, and attack-seq in single-cell level, and this is heavily led by Carla Jones and Anya Lorenk. Anya is here, so you can also look for her if you have more questions about that. So we actually had a training session in November 2022 because it was very important that all the sites were processing the data the samples in the same way. And this is very challenging because there's one thing of sharing just the protocol, but there are nuances in the protocol. I don't do wet lab, but every time I tried, I noticed this, like there is a tilt in the tube. There is something very nuanced that is not written in the protocol. So that's why we put everyone together, a person from each side, and they work together. They established the protocol. There was a lot of hands-on. And finally, we got a uniform protocol between all countries. It was super big work. We're also in communication with other large single-cell regional datasets like the Asian study, IDA. We use CPT tubes to lower the processing steps. And after blood collection, we process it immediately to lower the variability because of the weather. We have really hot weathers in our region, so that's also a challenge. And we are counting cells and viability before shipping and after being received at UK. So we really have a lot of controls. We have this double check of sample quality because we really want to develop the best possible data set for Latin America. We foresee that there are going to be very few data sets representing our region. We don't want this data to be low quality. We want it to be the best quality so it can be used in other research, so it can empower research for everyone. These are the controls for my lupus study. These could be the controls for your studies. They have to be perfect. So this is our current progress. Colombia has sampled 120 individuals. Mexico has also sampled the 120 individuals we have to. Peru is halfway. Chile is getting there to the end of the line. Argentina is really just waiting for people to finish their exams so they can keep sampling. But I know, and Marina, who is the hands in this, is here if you want to also talk about the progress in Argentina. And it has also taken a lot of time. So I told you that this actually started in 2022 in the training session. So it looks like, wow, what did you do in 2 years? No? But representation takes time. So one of the things is that standardizing the protocol took time, but also selecting healthy individuals with no chronic diseases, no recent infection, no mental health related episodes that were recent. It's very hard. People think they're healthy, most are not. And to gather the demographic and medical data from all participants also takes time. You need to convince people that they want to spend 20 minutes of their lives just answering your surveys. And we also want to have a very good coverage of age from young to 50 years old. And we also have to want to have gender balance. So at some point, for example, I know Peru is now going slower because getting men over 30 years old that want to participate in research is a challenge. And that's documented. That there are papers out there explaining how men are less likely to participate in research. So if you have friends that are healthy, that live in Peru, that live close to Lima, please point to Luis to visit them. One of the things I want to highlight is that we only have a restriction that the participants have to have both parents born in the country, but we don't really restrain for grandparents, but we ask where the grandparents came from. And you can see that most of our participants, their grandparents were also born in the country. So we foresee we will have a very high genetic diversity in our samples that will come from the native component. So we will see that very quickly because we actually just got first sequencing data for our participants, and hopefully we will show this to you next year. We also do the bucketing processing to enrich for cell types. This is very important because if you just plug PBMCs into single cell sequencing, you get a lot of T cells. Like everything is T cells. But as you know, my lab is very interested in dendritic cells, and we can actually get 5 different types of dendritic cells and 4 different types of monocytes. And this is a lot of work from Carla Jones and from Taran that have set up these pipelines. And again, like we also get a lot of T cells, people of T cells, we don't discard them, but we get other cell types, which makes us very happy. And analyzing this level of data gets a big team as well. So we have our team that works on Ancestry with Bennington and Sarah. Our team is focused on CyTic with Felipe and Marina, and you can see them in their posters, A and B, so both days. We have the single cell RNA-seq team with Juliet, that's part of Jessic's lab. And you can also visit her in the poster. The single cell ATAC-seq team, that is Domingo and Evelia, and you can see them as well in their posters. And Alejandra Schaffer and Anya. Ale is going to present for the EQTL team. And I will just give you snippets of the pipelines we are developing because you need to go to their poster because there's a lot of details. Otherwise, this is going to be super long and we're not going to have the amazing cheers that Jessie prepared. So in the case of CyTic, actually Felipe is developing a new pipeline and a new tool because there was lack of a tool that was taking into account the diversity of the signals that were produced by CyTic of the different cell markers, and that it really didn't incorporate the knowledge of immune experts. So this is something that Felipe has been doing also with Marina and with Danilo. The CyTic pipeline, we're incorporating different tools. We have discovered that we need to change some of the quality controls from these tools to adapt to our data set. And this is work done by Diego Ramirez, and you can also see him in his poster. For the single cell RNA-seq part, Juliet and the team are working, comparing different differential gene expression methods that are being explored and set up also in the cluster so we can really try them right away. And also that integrate RNA-seq with CyTic data. So these two teams are working together all the time. And to test other tools that can incorporate biological questions that are very interesting to us because we want to know if there is different amount of cell proportion between different ancestries and populations. We also have the EQTL mapping that is mostly focused on Mexican individuals, where we are going also to use cell stimulation, and that is going to be compared across the different stimulation. And this is the PhD thesis from Alejandra Schaffer that will be presenting as well her poster. And again, in Jaguar, we're applying this communication with participants. So everyone that joins Jaguar is part of the research, and we keep communicating with them through social media, through our webpage, traditional media with radio, TV, because actually older people like this media, and we need them. We have a big challenge. We are 7 countries. Spanish is not the same. So we need to really feature our postings so that they really go into the correct population. But we also have 3 languages, Spanish, Portuguese, and English. And if you know anyone in Uruguay that wants to be part of the project, Uruguay is starting collection next month. In summary, launching patient registries is a challenge, but it's also very rewarding. High quality data of healthy control individuals is key to empower disease research across the continent. So again, these are going to be the controls for a lot of research. So we want them to be well done. We're generating high quality data to improve representation of Latin American diversity, and our project potentializes functional genomic studies in the Latin American population. Community engagement has strengthened our project, having a more significant social impact through science communication. And I want to acknowledge, again, this takes a lot of people. So all the collaborators, the lab, that is amazing, our technical support, rheumatologists, the patient communities, and funding agencies, because you need money to do things. And again, the patient communities that are very important for us. Thank you for your time.
Thank you, Alejandra, for your talk. So it's time for questions.
Hi, thank you for the talk. It was great, and it's great to see this effort to have more samples in Latin America. I was wondering if there's any plan of including any epigenomic data in the registry.
So we're doing a taxic for Jaguar. At the moment, it's not that we don't want to. It's that it's expensive. But yeah, that would be like I'm writing grants all the time. But that's the idea of having the biobank. That whenever we get the money, we can start doing like generating other types of data.
Thank you very much. It was very interesting to see these initiatives, especially in Latin America. And so I had 2 questions, very unrelated. First one is about the funding, because there's a lot of work behind and it has to be funded. So is it, I'm sorry, but I'm not aware of how easy it is to write like joint grants between Latin American countries. Is it what you did, and how to do it?
So it depends on the project. But so, for example, for the Jaguar project, it was a network that already existed. Like we knew each other. So these conferences are great. Do talk a lot, make friends, start building common interests, and that's a very good way to start. I think we grew it like in like 1 month or something. It was very quick, and it was really amazing because the UK team had like this time difference, no? So you could see everyone, like there was always someone writing. Oh, it was amazing. So yeah, I don't think that it's hard. I know we have written like other grants, like with Vinicius, we tried something with the OECD, like to try to build like a bioinformatics institute in Latin America. It was not successful, but it's not like we cannot. We just need to keep trying. Like it's 1 in every 10 grants, no? That's the current perception of success. So for other grants, like some are based in Mexico from CONACYT, but for example, in the CONACYT grant, Gosha and Maria Gutierrez in Boston were already collaborators. So you keep building networks, and yeah, it works. You just need to keep writing grants.
Okay, thank you. And the second question has nothing to do with it. Is so I have my own interpretation why men volunteer less for these things than others, but I haven't read the paper. So could you tell us more about why is this sort of unbalance on
Yeah, there is the documentation that is a bit this perception of community advantages. So women are more interested in working towards the community benefit. Men are a bit more towards the individual benefit, and definitely spending 30 minutes answering surveys and being punched to get blood, it's not a lot of individual yeah, rewarding, but it's a community thing. So that's what the articles say, that is more this community perception of like who will get the advantage of this time I take to do this. Sorry, just after Javier, there is a question up there. Yeah, yeah, sorry. Yeah. No, it's fine, like, because it's going to take time to get there.
Thank you. Very nice talk. Two questions. One question is about the screening of population. You know that in very large genomic exercise of screening for population to get data from people, in Europe we commit some mistakes because, for example, in one case, 80% of the people that voluntarily went to these genomic projects, for example, in a famous project in Europe, where 80% were university degree people. And this is something that now in Europe we are trying to resolve, because you are not really doing a good search if your population is like that. No? This is one question. And another question is about dendritic cells. You know that they are very difficult to find and isolate. So you are producing dendritic cells derived from monocytes. This is the, let's say, the myeloid origin dendritic cell. What about the lymphoid origin dendritic cells that are also exist? And if you produce dendritic cells from monocytes, how different are they from macrophages? This is a dual question.
Yeah, so that we did, and they were there. There were different signatures. With actually, it was never published. There is a preprint, but the person that did let the work left. So we only have the preprint, but we could see that there was a clear difference. So it's while it's the same lineage, they are clearly the monocyte, the macrophage, and dendritic cells, and they have their own signature. And yeah, we are losing the other, yeah, the diversity of the other type of dendritic cells because of how we are doing this. But we really needed a lot of dendritic cells, and at the time we really didn't have like the power to do single cell analysis. But with the bucketing strategy, if we implement that with the lupus samples, we will be able to see a bit of that. Obviously not super rich, but would be present. And we know that we have that bias towards university people because we're based in universities. So whatever we do postings on social media, like at the end, the people that come are the people that are geographically closer because they can. It's not going to cost them a lot of extra time. There are people that eventually come from far away, but we know that's not that much people. We make an effort to get people out from the universities, so we go to blood banks. There's other strategies to go to gyms because usually that's healthy people, or to clubs, to like people that get together, like the churches. Like we are really trying to lower that bias, but we know it's going to be there because we are based in universities. Yeah.
Thank you for the talk. It's really nice to see this initiative here in Latin America.
I was wondering, since you mentioned that project is focused on single cells and gene expression, and that you host whole genome data, guys are interested to do genomic analysis of mutations, especially the ones on the major histocompatibility context where there is evidence of genetic mutations related with lupus.
Yeah, so we have we're currently sequencing using blended genomes. That means that we have 30x coverage for exome and 3x coverage for non-coding genomes, which is not enough for rare variants, but well, for the exome part, we can do that. We are hoping to get funds to do long read sequencing, for example, because the major compatibility complex is just going to be better with long reads. We'll have for that money.
Okay.
Hi. Thanks for the really nice talk. I have like many questions because I'm just I'm excited about how much data you were able to generate. The first one is just when is the data when the data is going to be available and how and like to what extent we're going to be able to manipulate the data on our own without that's okay. Then the second one what is the number you're gonna feel confident with to start doing the study? Because you're saying that you're collecting patients, right?
Mhm.
To use as control. But then like I guess that what is the estimate number that you're gonna need to reach to think that it's gonna be enough to start doing the proper research? And the third one,
I can start answering that part because I'm going to forget them. Sorry. I already forgot the first one.
how to
Data availability, yeah. So we are committed to like open the data set, obviously. The full data generation is going to be ending at the end of next year, which means that we still need time to generate our first analysis and obviously our own article. But after that, the idea is that the data is going to be available to everyone. Uh, there are some things. CZI, that is the one that is funding Jaguar, is very interested in having resources, like ways to really analyze this data. As you said, like from scratch. What if I want to analyze this, like from not really like count matrices, but something before? But that requires certain type of infrastructure because you need security for the data because genomic data is very personal. So we know that the count matrices will be available in this Cell Exchange platform. That's mandatory, like we have to do it. We can put the data on dbGaP, but again, that means you will need to have and prove to dbGaP that you have the infrastructure with the security to be able to handle that data. And it's a lot of data, so that's a big computer. But there are cloud resources that should be able to handle these, so you don't have to really go through all the bureaucracy and all the like the next level of ethics. And I think that's something CZI wants to fund, like to be able to. That's something that already happens in the Global Parkinson Genetics Project, where everyone accesses in one point in the cloud and they paid for the developing countries. So for example, my team works in that platform and I don't have to pay for that, but I also don't have to fight to get the data for Parkinson patients in Asia. It's already there. I can read it, I can use it for my analysis, but it gets me out of the like bureaucratic steps to get the data into Mexico and to have the storage capability to host it. So I think that's something that he said I would like to fund. So like again, the data is going to be there, but what happens when the data is there and we don't have the computing power to like that's again leaving us stranded. And again, the people that are going to take advantage of that are the first world country places that have the capacity to deal with the data. So yeah, lots of discussions there. And then second, yeah, sorry,
In
second
terms of the
question.
number
Yeah,
patients,
so I did a power calculation once with a friend for another grant, but it's 200. Like you can get EQTLs with 200 samples. So that's the short answer. I don't remember exactly how we did it. I have a paragraph that describes what we did. It was like a year ago. But yeah, 200 samples is fine.
so and also, do you think that one of the reasons for not having enough patients in some countries is that probably they haven't given enough information about how their data is going to be managed, like in terms of privacy, and how it's going to be kept secret, like from being decoded for different purposes other than research?
So I know that would be the case for Mexico. But I don't know for other like yeah, the perception from the other teams might be more useful in this case. Because I know in some cases is because of the university's structure is very hard to reach out from the university. And again, as Javier was noting, we don't want just to sample the university. So yeah, it really depends on the site, but it also depends on the local ethics regulating the project. So yeah, maybe talk to each team to know what's happening in each country will be easier.
Thanks.
Thank you, Alejandra, for that excellent talk. My question is, what type of metadata are you collecting from those healthy controls? What I want to know at what extent are you able to take all the metadata that is required for further studies in immune response? Because immune response you need to have nutritional data, stress data, previous vaccine data, previous infection exposures, and a lot of things.
So we do a very thorough medical survey with all like the medical history of disease, if they broke a bone, just depend on what people can remember. But we do that. It takes about 15 to 20 minutes. And we also, and that's one of the things, like some surveys are optional for people just because it will be too much time. And we send them afterwards through email so they can answer them later. There is an issue with dropout. Like at some point, people get tired and they just stop answering the surveys. But we are aiming to collect as well mental health status. Is a very general survey, but also sleep patterns and eating patterns as well, like how they eat. But yeah, depending on the survey, maybe less people answered the latest one. But the medical survey, we have it completed for everyone.
Okay, and my second question is, what is your definition for healthy?
So it will be in general people with no chronic illnesses, no recent infections, and no recent mental health issue episodes, like no recent intervention because they had a very big depression episode or an anxiety attack in the last 6 months.
About comorbidities.
So they cannot have anything that is chronic. So they cannot have diabetes. They cannot, like, yeah, any chronic thing, it's a no. We usually try to focus in blood bank recruitments because usually those people already have most of our requirements.
Thank you so much.
Yes, I have another question related to the metadata.
Mhm.
It's related to if
you have considered the enrollment of the populations where the adolescents came from? Because as I know, here in Colombia, at least in Antioquia, Boyacá, and in the Caribbean coast, there are highly enrollment populations, and this can be affected by consumption, right? So have you also considered that kind of mentality?
So that people are related? That's the question?
Yes, yes, so they agree.
Ah, so we think we have a very broad recruitment process, so that shouldn't be an issue. But we are also doing whole genome sequencing, and we check if they are related. Up to now, no one has come to be related. So yeah, we hope not.